Welcome to this explainer. For the longest time we've looked at the death of a cell as a failure, just a biological machine breaking down under the wear and tear of life. But today, we're going to completely shatter that idea. Modern molecular biology has shown us that cell death isn't just some morbid, accidental tragedy. It's actually a highly orchestrated, absolutely essential feature of life itself. It's built right into our biological architecture, and understanding it, well, that's unlocking some of the most mind-blowing advancements in precision medicine today.
Let's dive right in with a fascinating, slightly mind-bending question about the very hands you're using right now. Have you ever actually wondered how your fingers formed? Here's the crazy part. Before you were even born, millions of your own cells willingly committed suicide to shape those hands. No accident, no disease. It was a perfectly timed, highly organized cellular sacrifice. So why did they do it? And more importantly, who or what gave the order? To answer that, here is our cognitive roadmap for today's explainer. First, life's strategic choice of death. Second, the history of programmed death. Third, why versus how we die. Fourth, accidental or regulated death. And finally, the era of treatable death.
All right, Section 1, Life's Strategic Choice of Death.
In evolutionary terms, cells don't just die because they give out. They literally eliminate themselves for the survival, efficiency, and perfection of the entire living system. Think about those hands again. This perfectly explains why you have individual fingers instead of webbed hands. The cells between your developing fingers essentially agreed to die on a very strict schedule. This process is called fetal digit formation. If those cells hadn't self-destructed at exactly the right biological moment, your hands would look completely different. So this elimination isn't random at all. It's an incredibly precise developmental mechanism.
There is a fantastic quote that sums this up perfectly. Death is not a byproduct of life, but a part of the blueprint that completes it. The crucial takeaway here is that this organized cellular sacrifice is literally the fundamental blueprint of life itself. The death completes the design. We almost always focus on growth, cells multiplying to build a body. But the removal of cells is just as vital. Think of it like a sculptor chiseling away at a block of marble. The art isn't just created by the stone that remains, it's created by the stone that is systematically taken away.
Let's move right along to Section 2, History of Programmed Death.
It's just incredible to trace how our understanding of this shifted over the decades. Back in the 1960s, researchers Richard Lockshin and Carol Williams were simply observing insects. They noticed that certain muscles degenerated at highly specific times during metamorphosis. The timing was just way too perfect to be random. Fast forward to the 1990s, and scientists actually pinpointed the exact genes controlling this process. And today, in the modern era, we don't just see a simple on and off switch. We map out massive, complex networks of interconnected cell death pathways.
That huge breakthrough in the 90s actually came from studying a microscopic worm called C. elegans. By looking at these worms, scientists realized, amazingly, that they could literally flip a genetic switch to alter a cell's ultimate destiny. C. elegans was the absolute perfect model organism because its development is 100% predictable. Researchers found that specific genes, namely SEED3, SEED4, and SEED9, were completely responsible for dictating whether a cell lived or died. This was a monumental discovery. It proved that death wasn't just bad luck. It was a biological command written right into the DNA.
Which brings us to a major paradigm shift in Section 3, why versus how we die. You might hear these scientific acronyms mixed up sometimes, but it's crucial to keep them straight. Apoptosis is the how. It's merely the execution mechanism, focusing on the physical features, like the cell shrinking and condensing as it dies. On the other hand, programmed cell death, or PCD, is the why. It's the underlying biological motive, like a strict developmental schedule. So, for example, if a cell dies from severe external stress, it might use apoptosis to execute that death, but because it wasn't part of a natural developmental schedule, it's not technically PCD. Understanding this distinction is absolutely key to navigating modern cell biology.
So what actually happens when cells die for the right, scheduled reasons? Well, programmed cell death performs some incredibly essential tasks in humans. We already talked about fetal digit separation, but it also optimizes our nervous system. Early on, our bodies overproduce neurons, and PCD carefully prunes away the excess to create streamlined, efficient brain wiring. It also maintains our immune system homeostasis by removing rogue lymphocytes that might mistakenly attack our own organs, which prevents autoimmune diseases. Basically, it's a brilliant built-in quality control system.
All right, Section 4, accidental or regulated death.
This introduces the most critical dividing line in modern cell biology. There is a massive difference between an unavoidable cellular car crash and a highly choreographed exit strategy. Accidental cell death, or ACD, is the car crash. It happens through immediate, extreme physical or chemical trauma, like severe burns. The cell is instantly destroyed, and it's totally impossible to control. But regulated cell death, or RCD, relies on systematic molecular pathways. And because it uses a pathway, it is controllable. Forms of death like apoptosis or ferroptosis fall right into this RCD category.
Because of this, modern researchers use a very specific, three-step investigative framework to solve the mystery of every dying cell they encounter under the microscope. Step 1. Why? They assess the physiological or pathological context. Step 2. Is it regulated? They figure out that critical binary threshold. Is it RCD or ACD? And finally, Step 3. How? They identify the exact execution tool being used, like necroptopsis. This precise framework is what guides today's cutting-edge therapies.
Which leads us perfectly into Section 5, the era of treatable death.
This is where the theory turns into a high-stakes medical revolution. The guiding principle here is simple but profound. Regulated death is treatable death. It is a deeply empowering realization. If a cell's death follows molecular rules, well, we can rewrite those rules to cure devastating diseases. If regulated cell death operates on biological pathways, that means it has switches. And if it has switches, medical science can design drugs to either turn those switches off or force them back on.
This gives us the incredible dual strategy of precision medicine. On one side, you have cancer. Cancer cells are notorious because they've mutated to resist natural death pathways. They essentially refuse to die. So the goal is to reactivate those dormant apoptosis pathways and force the immortal tumors to finally self-destruct. On the flip side, you have ischemic diseases, like a stroke. During a stroke, healthy brain cells panic and trigger an overactive death pathway, like necroptosis. In that case, we want to inhibit those specific pathways to halt the damage and save that healthy tissue. It's a total game changer.
And we are getting incredibly precise with this. Take ferroptosis, for example, a recently discovered form of iron-dependent regulated cell death. Scientists have found highly specific druggable targets, things like GPX4, FSP1, SystemXC, CoQ10, GCH1BH4, and DHODH. Now, you absolutely do not need to memorize that alphabet soup. Just think of them as volume knobs on a biological soundboard that control things like lipid peroxidation and iron metabolism. By inventing therapeutics that target these specific switches, doctors can actively engineer a cell's sensitivity to death, dialing it up or dialing it down.
We have literally moved from seeing death as this mysterious, unavoidable decay to recognizing it as a precise, druggable network of biological code. We are entering an era where we hold the blueprints to life's most dramatic exit strategies right in our hands. As we map out these complex networks of cellular fate, we're left to ponder one final, provocative thought about the future of human medicine. If we can actively engineer a cell's death, exactly how far can we push the boundaries of life? Thank you so much for joining me on this explainer today. Keep questioning, keep learning, and I'll catch you next time.